All right, let's start. Is Godfred here? Godfred.
Yeah, doctor, I'm here.
Yeah. Are you less busy now?
I am driving home now. I just closed from church.
Okay, okay, that's fine. All
But
right.
I'm in class.
So we are looking at the cardiovascular system, and I'm sure you are doing the anatomy part. Okay. I'm aware you are doing the anatomy part. So that means our class will be a little easier compared to not doing the anatomy part. So you are saying the cardiovascular system is made of the heart as an organ, and then we have blood vessels. And we want to look at the functions of these components, and then how blood pressure is maintained, how the cardiovascular system is regulated, and how this same system is regulated under normal physiological conditions. So this is the first part of it. Hopefully next semester you continue with the second part. And I think we have just this today and the next Sunday to end the semester. So let's try and then pay attention and then be attentive so that we can move faster. So with cardiovascular physiology 1, we want to look at the overview and the functions of the system. We identify the primary functions of the components. We have blood, and these components, we have blood vessels, the heart, and so on. We also identify some proteins and other solids that is found in the blood and what they do. Then we'll also look at the functions of the cellular components of the blood. Talk about red blood cells, white blood cells, platelets. We look at hemoglobin and its function and why it's very important. Then we also classify white blood cells and then their unique functions that they have. Then we also look at blood grouping systems, and then we look at blood transfusion and blood transfusion compatibility and so on. Then we also look at the occurrence of erythroblastosis fetalis, which is the hemolytic disease of the newborn. So basically that's physiology 1. We end there, then we continue with physiology 2, where we look at the conducting system of the heart and regulating blood pressure and so on. So just as you've started the anatomy part, I'm sure you are aware of the components of the circulatory or cardiovascular system. We are saying it's a closed system. It's a closed system, meaning the heart pumps blood throughout the body. There's no open ends. Okay, so the blood circulates throughout the body and returns back to the heart. So there's no end of blood circulation at one point. Blood circulation is done, and then blood returns back to the heart. We are saying this system is made of the heart, the blood, and then the blood vessels. So we are saying the heart functions to pump blood, and blood vessels will carry blood throughout all parts of the body. So we are saying the heart is an organ. Let me ask this from the class. What is the blood? If the heart is an organ, blood vessels are organs. What is the blood? Any idea? Not even any idea. This one you should all know. Okay, so just can someone answer it so that we move on faster?
May the question
Yes.
again, please?
Okay, what is the blood? If the heart and blood vessels are organs. I saw Martin, right?
The blood is a connective tissue.
Yes, the blood is a connective tissue. So it's made of some cells, and then we have the extracellular matrix that gives it the structure of the connective tissue. Okay, so we are saying the function of the cardiovascular system as a whole is to transport oxygen and nutrients, and also remove carbon dioxide and other waste products from the body. So if we're doing homeostasis, I made mention that the main extracellular fluid that maintains homeostasis is the blood. Okay, because it's the blood that transports heat, nutrients, oxygen, other vital components that the body needs, every cell needs. It's the blood that supplies it. And then we're also saying it's the blood that moves waste products, okay, transports waste, urea, creatine, and all these metabolic waste is released into the blood where they travel to the kidney to be filtered. So the blood is very, very important. It's a very, very important extracellular fluid that helps the body. So you are saying the regulation of cardiovascular system occurs via an innumerable number of stimuli. You are saying including changing blood volume. So we always and constantly regulate the cardiovascular system. This is because there are changes that occurs severally. For example, the food we take in, water, other electrolytes, hormones that are released, and so on, concentration of the blood, medications we take, and all these changes the pressure, changes the volume, changes the concentration of the cardiovascular system, mainly the blood. Okay. And we are saying we need to regulate this system so that it doesn't also affect other systems. Because we are saying the blood is what transports almost everything that every cell needs to the cells and tissues. We are saying the parasympathetic and sympathetic nervous systems play a key role in regulating the cardiovascular system. So if we are to increase your heart rate or decrease your heart rate, depending on the circumstance, we are saying is the nervous system that acts or that plays that role. So the nervous system, as I said, controls every system, including the cardiovascular system. So when you take the heart as an organ, we are saying that is the main organ of the cardiovascular system. And it's found at the thorax, between the lungs, and its pointed apex is directed towards the left hip. And all this is anatomy, I'm sure you've done it. It's about the size of your fist. The main function of this organ is to pump blood through vessels. They are saying it pumps blood directly into arteries, more specifically the aorta or the pulmonary artery. So we can classify the function of the heart in four main parts. The first one, specifically, we are pumping oxygenated blood to the other body organs. When you say oxygenated blood, meaning we are saying this blood contains or is highly saturated with oxygen. It's highly saturated with oxygen. We are saying this same heart pumps nutrients or blood containing nutrients and other vital substances to different organs. This same heart also receives deoxygenated blood, that is blood that is less saturated with oxygen. Okay, so you have less saturation of oxygen in deoxygenated blood that travels to the heart and is pumped to the hands to receive oxygen. So another function of the heart is to maintain blood pressure. The force or the pump the heart being a pump generates the force that pushes blood to every part of the body. So that contraction of the heart muscles generates the pressure that forces blood to move to every part of the body. We're going to look at the pathway of blood through the heart. The pathway of blood through the heart. When you say the pathway, we are looking at how blood will travel from the receiving end of the heart to when it's leaving the heart to the body. So we are saying that, kindly follow the pointer. We are saying that we have deoxygenated blood returning back to the heart, okay, from the body. The body has used up the oxygen, and this deoxygenated blood is returning back to the heart. So we have the blood from the lower part of the body returning through the inferior vena cava. We have the blood returning from the upper body, returning from the superior vena cava. So this blood enters the right atrium, Then it flows down into the right ventricle. Now this blood also moves or is pumped through the pulmonary artery to the left lung or to the lung. When it gets to the lung, there's exchange of oxygen and carbon dioxide components. Oxygen diffuses into the blood, carbon dioxide leaves the blood into the alveoli. So when the blood is returning from the lungs, that is where it's highly saturated with oxygen. Now this oxygenated blood enters the heart through the pulmonary veins. It enters the left atrium, then it flows into the left ventricle. Then we have the blood from the left ventricle leaving the heart through the aorta. Now the aorta branches into other arteries that supplies this oxygenated blood to every part of the body. So can someone, just as I did, describe the pathway of blood flow through the heart? Anyone? Just one submission, we move on. Irene.
Okay, sister, I want to try.
Okay.
So we said that deoxygenated blood, that is blood which has gone through the system and oxygen in it has been used.
Yeah.
Enter back into the heart through the superior vena cava, that is the upper part of the blood. The deoxygenated blood enter through the superior vena cava.
Okay.
And the deoxygenated blood at the lower side enter through the inferior vena cava into the right atrium. So from the right atrium, the blood is moved to the left ventricle. From the left ventricle, it is moved through the pulmonary vein, the left and right pulmonary.
Okay, let me correct
vein.
you on that.
Sorry, pulmonary
Irene.
artery.
Hello, Irene.
Sister.
Yes, so
Yes,
when
sister.
the blood moves from the right atrium, it moves to the right ventricle. Okay.
Yes.
So it's the right. I heard you saying left. I don't know if I'm the only person, but I'm sure.
No, I said left and right pulmonary artery. That is how it leaves the right atrium, ventricle, sorry.
Okay.
So from the left ventricle, it goes out through the left and then right pulmonary artery into the lungs.
No, Irene, you see, this is the left ventricle. Whenever you mention left ventricle, that means it's oxygenated blood that is leaving the heart to the body. Okay, so
sister, please, I'm not there yet, please.
Yes, but you said left ventricle.
I said left and right artery. Like where it's going to the lung.
Can you start over?
Let me start over.
Oh, sure.
Please let me start over. He said that deoxygenated blood in the body enter the heart through the upper part enter the superior vena cava, and then the lower part enter through the inferior vena cava into the right atrium. So from the right atrium, it moves to the right ventricle.
Okay.
Then from the right ventricle, it passed through the left pulmonary artery and then the right pulmonary artery
Okay.
into the lungs, where
Good.
it is then oxygenated or mixed with oxygen.
Okay.
Now the oxygenated blood now enters back into the heart through the left pulmonary vein into the left atrium. Then it moves from the left atrium to the left ventricle.
Okay.
Then from the left ventricle, it moves through the aorta
Okay.
into the body.
Great. Great. Well done. Well done. That is it. Yes. Let me take the last, just one person. Atosa. Atosa.
Okay. Saying that deoxygenated blood returns to the heart by way of the superior and inferior vena cava. So the blood will now get to the right atrium. Then from the right atrium, it will get down to the right ventricle. Then from the right ventricle, the blood is pumped into the pulmonary trunk. And the pulmonary trunk, as it keeps on moving, it branches into the left and the right pulmonary arteries. Then it gets to the lungs.
Okay.
When it gets to the lungs, it unloads carbon dioxide and takes oxygen. So now the blood will now return back to the left atrium by way of 4 pulmonary veins, 2 from each of the lungs. Then when it gets to the lungs, the blood is now moved to the left ventricle. Then it's pumped into the aorta, and for that matter, the systemic circulation.
Great. Great. Well done. Well done. All right. So let's move on. That means you get what I'm saying. So this just a representation of what I'm talking about. Okay. So you have the blood from the upper and lower extremities coming through the right atrium to the right ventricle to the pulmonary arteries to the lungs. Returns from the lungs to the left atrium to the left ventricle. Blood is pumped out the body through the aorta, branching into different arteries. We want to look at the types of blood circulation. We have systemic circulation where we have blood leaving the heart to every part of the body, every part of the body. So with this systemic circulation, we are saying the main chamber where the blood is pumped out is the left ventricle. Okay, the left ventricle. So blood leaving the left ventricle would be pumped through the aorta to the system, to every system. They're saying 85% of this circulation is moving to every part of the body. We have 5% remaining in the heart, and then we are saying this systemic circulation is under high pressure. It's under high pressure. Then we have pulmonary circulation, or lung circulation, where we have blood pumped to the lungs. And you are saying is the right ventricle, that is the main chamber of the heart, that pumps the blood to the lungs. And this is done under low pressure. We have 10% of circulation moving to the lungs. Then we have coronary circulation, where we have blood leaving the left ventricle directly going to the heart muscles. Okay, so it leaves the left ventricle, passes through the aorta, passes through the coronary arteries back to the muscles of the heart for oxygenation. So we are saying pulmonary circulation allows for oxygenation of the blood. Pulmonary circulation is blood moving to the lungs, and therefore when it gets to the lungs, it takes in oxygen, or it exchanges its carbon dioxide component with the oxygen component in the alveoli. Therefore, we are saying pulmonary circulation allows for oxygenation of the blood. Systemic circulation provides the body systems with oxygen and nutrients that it needs. So in coronary circulation, you are saying blood in the heart chambers does not nourish the myocardium. So you are saying as blood is moving through the pathway that you've just mentioned, or you just described, it doesn't exchange, or it doesn't nourish the muscles of the heart. It doesn't. Okay, so we are saying the heart itself also needs a channel through which there will be supply of blood to supply of blood, oxygen, nutrients for the muscles, for the cells to survive, to function properly. So you are saying the heart has its own nourishing circulatory system called the coronary circulation. And it's made of the coronary arteries, the cardiac veins. And you are saying blood empties into the right atrium via the coronary sinus. So you are saying we have the arteries, coronary arteries and veins that supplies blood and returns blood around the heart. Okay, so it supplies blood to the heart muscles itself, supplying oxygen, nutrients, and other vital substances every cell, tissues, and organ would need. So the blood passing through the chambers doesn't do that. We also need blood supply to this heart as an organ, to the muscles there, to the tissues there, to the cells there as an organ. We have blood vessels. Blood vessels are they are channels through which blood moves from the heart to every part of the body. And we have arteries, we have veins, we have capillaries. So we are saying arteries, they carry oxygenated blood from the heart to every part of the body. And then we have veins also carrying deoxygenated blood from every part of the body back to the heart. So mainly we are saying arteries carries oxygenated blood away from the heart. And it's 3-cell layered, has a thick wall compared to the veins and capillaries. We are saying with the veins, we have valves that prevent the backflow due to the low blood pressure in the veins. And it's also 3-cell layered, thin wall, sorry. Then we have capillaries that are connected. They are serving as a connection between the arteries and the veins. So at that tissue level where we have exchange of oxygen, the nutrients and other vital substances with the tissues, is the capillaries that exchange the blood component with the tissues and cells. And you're saying the site of capillaries or tissues is where nutrients and waste exchange by diffusion takes place. And there's 1-cell layered, very thin, to make sure there's easy exchange from the blood and then to the cells and tissues. So to look at the other differentiations among the three, we are saying the artery is elastic compared to the vein. Elastic meaning it has elastic fibers or proteins that enables it to stretch to accommodate blood under high pressure. Okay, so it stretches during blood flow so that it can enable, or it can allow for high pressure during blood circulation. Veins are less elastic, and then with capillaries, there's no elasticity. And then with the artery, we are saying it has a narrow lumen compared to the vein. And then we are saying with capillaries, there's also very narrow lumen. And due to that, we are saying the pressure inside the artery is high. And of course, it's also carrying pressure from the main pump, which is the heart. With vein, since the lumen is large, the pressure is low. And then with capillarity, it's very narrow, and therefore the pressure there is also low. Arteries, they don't have any valves. Veins, they have valves to prevent that flow of blood since there is low pressure in veins. Capillaries, they don't have any valve. Looking at the main functions of the cardiovascular system, the first one, transport of nutrients, hormones, and other vital substances cells need. Removal of waste. We are saying gaseous exchange, that is transporting carbon dioxide out of the body through the alveoli, the lungs, out of the system. And then we're also saying oxygen diffusing into the blood, and the blood taking this oxygen to the tissues and cells. Immunity, we are saying we have some components of the blood that protect the system. Mainly, we have the white blood cells that fight against infections, diseases, pathogens, any foreign material that comes inside the body. So it provides immunity to the body. We are saying blood vessels transport blood, and this blood carries oxygen, carbon dioxide, nutrients, waste, and all these substances that is essential for the cells to survive. The heart pumps blood through blood vessels. We look at the blood. We are saying blood is a fluid connective tissue. Connective tissue, we already know why it's a connective tissue, and it's the most crucial component of the circulatory system. Very, very crucial, very important. We are saying in a healthy person weighing 70 kilograms, the total volume of blood is approximately 5.5 liters, circulating throughout the person's body. And this blood is made of four elements, which are cells and cell fragments. And then they are suspended in a liquid fraction called plasma. The function of blood is transportation, oxygen, carbon dioxide, nutrients, waste, etc. Then we have regulation of temperature, pH, osmotic pressure, and so on. So blood helps regulate body temperature by excreting excessive heat during when you are in a hot environment. We just did that last week, homeostasis. When you're in a cold environment, it's this same blood that needs to maintain the heat within it too, so that you can maintain the normal body temperature. With osmotic pressure, we are saying it occurs when, for example, you try to maintain the normal volume of water or the normal plasma level. When you do that, maintain blood pressure, normal blood pressure. And then with pH, we are saying we have the normal pH range. We talked about it the last time. And therefore, it's this same blood that serves as a buffer system to maintain how acidic or alkaline the blood should be. The blood pH is slightly alkaline, and the blood has its ways and means of maintaining this alkaline state to help the cells and tissues to survive, to function well. Then protection. We are saying we have white blood cells, as I mentioned earlier, that protects the body from infections, diseases, as well as we have the cell fragments or the platelets that help in clotting. Aside that, we have clotting factors, which are proteins that is found in the blood that help in preventing excessive loss of blood when bleeding. Okay, so these are the functions of blood that we can look out for. Looking at the composition of blood, we are saying it's made up of 8% of normal body weight. So if you weigh, let's say 70 kilograms, 8% of this weight is made of blood. Then we are saying when you take 100% blood, we are saying 55% is made of plasma. We are saying plasma is the fluid part. Okay, the fluid part. Then the remaining 45% is made of cells. And these cells are red blood cells, white blood cells, and then the platelets. But mainly, the 45% is significantly red blood cells. So the 45% is mainly red blood cells because the white blood cells and platelets is less than 1%. Okay, it's less than 1% in a healthy individual. It's when there are infections and others that this white blood cells increase in percentage. And those who have worked in the lab before, when you centrifuge a whole blood, you get the next tube that's with the differentiations. Then you get it. Sometimes you don't see that whitish layer between the plasma and then the red blood cells. That whitish layer is called the Buffy coat. The Buffy coat is made of the white blood cells and platelets. So in a healthy individual, that layer is not significant. You barely see it. Okay. So we are saying mainly in a healthy person, whole blood is made of 55% plasma and 45% red blood cells. And you are saying hematocrit is the percentage of red blood cells you have in your system. So we are saying 99% of cells are called hematocrits. That means we have almost, or the significant part of the cells, mainly red blood cells. As I said, we have less than 1% making up white blood cells and platelets. Then we are saying the plasma is the fluid portion of the blood, and it's a solvent for electrolytes. And we also have proteins in there. We have hormones in there. We have other components where we have nutrients, waste, and all these are found in the plasma of the fluid part. So we are saying if whole blood is allowed to stand, clot is formed. It can clot. Okay, so when you remove clotting factors, so it can clot because you have clotting factors. We have some proteins that enables blood to clot in the plasma. So when you remove these clotting factors, the remaining fluid part is called the serum. So we are saying the serum is the same composition of plasma without fibrinogen, without prothrombin, without other clotting factors. We are about 12. So taking all these clotting factors out, you get serum. Then we look at the protein component of the blood. The protein component of the blood. So with the protein components, the main one that we all know of is the albumin. The albumin maintains oncotic pressure in the blood. This pressure prevents the fluid part of the blood from leaking into the tissue. So mainly it makes the capillary wall or the blood vessel wall impermeable to water, such that it will prevent the leakage of the fluid part of the blood into the tissues and then the cells. We are saying this helps move smaller molecules through the blood, including bilirubin and other hormones, vitamins, and so on. So it enables these molecules to be transported to where they are going to without them leaking into surrounding tissues and cells. And then we also say we have another protein called globulin. Globulin, we have alpha, beta, gamma, that is produced in the liver by the immune system, and it helps in fighting against infection. So it forms part of the immune system that is in protection of the body. Then we have fibrinogen and clotting factors, as I mentioned. They help prevent bleeding when your blood vessels are disrupted or exposed. Blood buffer system. Before that, any other question? Any other question? or any question? Class, are you here?
Yes, we are there.
Yes, we are there.
Yes, ma'am.
Okay. All right. So let's look at blood buffer system. As I said, this system helps maintain the alkaline pH of the blood. And this pH needs to be maintained. You did it during homeostasis because that is the pH that the cells function best. When the cells become acidic or more alkaline, it alters their function. The normal pH of the blood is slightly alkaline, 7.35 to 7.45. And this pH is supposed to be maintained. We are saying we have the bicarbonate or carbonic acid buffer. The bicarbonate or carbonic acid buffer. And we are saying this is abundant in the ECF. And here, as we are saying, the ECF is the blood. And then it functions almost instantaneously within seconds. How does this occur? We are saying when cells, red blood cells, pick up oxygen, the oxygen travels to cells, to tissues, and then it uses up the oxygen and produces carbon dioxide. This carbon dioxide would enter red blood cells and combine with water to form carbonic, which will also dissociate or break down to form the bicarbonate ion and then hydrogen ion. Now, bicarbonate is pumped out to the plasma. So this happens in the red blood cells. The bicarbonate moves into the plasma or the fluid part. That is what happens in the blood. So it's this bicarbonate, which is a base that maintains the slightly alkaline nature of the blood. However, when the blood is more alkaline, we have the release of the hydrogen ion to counteract that pH. At the alveoli level, we are saying bicarbonate will re-enter red blood cells, and the above equation is driven to the left, moving this to the chloride shift. So we are saying when it drives to the left, we are saying this reproduces carbon dioxide and then is eliminated by the lungs. So mainly what happens in the blood and why blood is a buffer system is when carbon dioxide diffuses into red blood cells, forming the carbonic acid. When that is formed, same within the red blood cells, this carbonic acid dissociates into hydrogen ions and bicarbonate ions. Now, it is this bicarbonate that would maintain the slightly alkaline environment of the blood. So, to give a summary or a summary of the brief description of the blood we've just learned, we are saying 55% of blood, whole blood is made of plasma, which is the fluid part. And within this, we have proteins, we have hormones, we have vitamins, we have nutrients, we have a whole lot, okay, that make up the plasma. But we are saying that 90% of this plasma is also water. 90%. So the components that we've mentioned so far makes up 10% of the plasma. And therefore, your intake of water has influence on the level of plasma, and therefore it has influence on the volume of blood. We are saying the red blood cells is the main or abundant cells in the blood, made of 45%. With this 45%, we are saying we have less than 1% made of white blood cells, platelets. So we want to pick up the plasma again. Already I've mentioned that it's 90% water. And water maintains the osmotic pressure in the blood. And we also have proteins. You mentioned the proteins. You have electrolytes, the different ionic components of the blood. And then aside that, you have the nutrients, the hormones, the waste, and then the gases. All these, that is found in the plasma. The purpose of the plasma is to transport their components to where they are needed. And we also have cells depositing their waste products in the plasma. You are saying the blood plasma also ushers the movement of all other elements of blood through the circulatory system. So mainly, the blood plasma, since it's a fluid, is what is transporting its components to every part of the body. Now we look at the cell components. With the cell components, the one that we said is abundant is the red blood cells. The red blood cell is also known as erythrocyte. We are saying it's a cell. We mentioned during introductory physiology that it's a specialized cell. It's a specialized cell because it's not having organelles. It's not having organelles, and then the shape is quite different from a generalized cell. You can see that the center is dented. Okay, so it's dented. It doesn't look like the normal cell. It has a cell membrane, right? And the color also is red. That is not similar to the general cell. We want to look at why the red blood cell is red, the function of the red blood cell, and why the center is even dented. Okay, so aside that, you are saying the cytoplasm is filled with a molecule called hemoglobin. And this hemoglobin is what makes the cell a red blood cell. Because we know that the red blood cell functions to transport oxygen into all cells tissues. And it's this red blood cell that also transports carbon dioxide out of the body. Now, this is done with the help of the protein called hemoglobin. Because this protein is what the oxygen and the carbon dioxide will bind to be transported. Functionally, red blood cells transport oxygen from the lungs to all parts of the body. As I said, we have modifications that makes it specialized. This red pigment that binds to oxygen, that is the hemoglobin. So it's the hemoglobin that gives the cell the red color. It's the hemoglobin that gives the cell the red color. And its shape is also not generalized. It has a biconcave shape. And this shape increases surface area to volume ratio. That enables diffusion of oxygen to be faster. And the cell membrane is also flexible. And it allows red blood cell to bend, to squeeze through narrow capillaries. So it's not rigid, it's very flexible so that it can pass through capillaries and arteries, veins, whichever blood vessel it finds itself in. They are saying, according to Ball's effect, hemoglobin has a lower affinity for oxygen. All that we are trying to say is that when you have oxygen and carbon dioxide in a solution in the presence of red blood cells, hemoglobin will bind to carbon dioxide rather than oxygen. Hemoglobin will bind to carbon dioxide rather than oxygen. Therefore, when you find yourself in a polluted environment with an atmosphere made of carbon monoxide, carbon dioxide, all these waste gases, or yes, these gases, we are saying red blood cells, as air enters your lungs, red blood cells will pick up carbon dioxide instead of the oxygen you have. So according to Ball's effect, we are saying hemoglobin has lower affinity for oxygen compared to carbon dioxide. Let's look at hemoglobin. Hemoglobin, we are saying, is made of 4 strands of amino acids, and it binds fully to all 4 strands. And we are saying hemoglobin is 100% saturated with oxygen when 4 molecules of oxygen binds to these 4 strands of amino acids. And with these 4 strands of amino acids, each of them has a binding site made of iron. So this iron is where oxygen binds to. So we are saying that hemoglobin is made of 4 strands of amino acids, and each strand is made of an ion as a binding site for oxygen. So when you take 1 hemoglobin, it will be able to transport 4 molecules of oxygen. 4 molecules of oxygen. All right. So we are saying that at the alveoli level, we have oxygen diffusing from the alveoli sac into the capillary, where we have the blood cells or the blood. And we also have the exchange of carbon dioxide from the blood into the lungs. At the tissue level too, we are saying oxygenated blood carried by the artery moves to the tissue or cell where there is diffusion of gases, there is transport of nutrients and other substances from the blood into the tissue or cells at the capillary level. Then, as transport is done, we are saying we have the remaining components, and where there's diffusion of carbon dioxide and waste into the blood at the venous level or getting to the vein. Okay, so we are saying at the capillary level, we have the arterial level and then the venous end. So between these ends, we have exchange of nutrients and oxygen, other components at this level. White blood cells. We are saying white blood cells are also known as leukocytes. These leukocytes are found in the immune system. So they are immune cells. They help defend the body against infections, against foreign materials, pathogens, anything that the body recognizes as foreign. And we are saying that we have several different types of white blood cells. And they share some common structure and then some function. Then we also have some in their distinct form, both in structure and in function. We are saying white blood cells are produced in the bone marrow together with other cells, red blood cells and white blood cells. All these cells are produced in the bone marrow by the hematopoietic stem cells. And these stem cells, whenever you hear stem cell, we have other cells that will come out of these stem cells. So it's this stem cell that will differentiate into different cells. So it's this hematopoietic stem cell that will differentiate into white blood cells, and then red blood cells, and then platelets. But mainly, this hematopoietic stem cell differentiates into the lymphoid progenitor cells and then the myeloid progenitor cells. I'll talk more about this in maybe next 2 slides or so. We are saying a major distinguishing feature of these white blood cells is the presence of granules. And some of them contain these granules, and there's absence of these granules. We are saying white blood cells are often characterized as granulocytes or agranulocytes. So granulocytes meaning they contain these granules. A granulocyte is without granules. So we have different types of white blood cells. Class is 2, quiet. So from the slide, let's discuss the functions of the white blood cells. And from what you know, class, I hope you are with me.
Yes, madam.
Yes, Daddy. We are there.
Yes, please. We are with you.
Class is very quiet.
madam.
Okay. Let's have a discussion on this. The types of white blood cells and their function. Yes, Irene.
Yes, madam.
Irene, are you answering the question?
No, madam, please.
Okay, anyone? Yes, Rose.
Okay, madam, please, from the slide, we have two types of white blood cells, which are the granular leukocytes and then the agranular leukocytes. And then under the granular leukocytes, we have the neutrophils, which fight against bacteria and viruses. The eosinophils, which play their role in allergy symptoms. And then the basophils, which also play their role in asthma. Thank you.
Okay. Any other? Any other? Hey. Irene.
Okay, sister. According to the slide, the agranular leukocytes, they said they are also divided into two parts. That is the lymphocytes and then the monocytes. And they said the lymphocytes they are the antibodies producing cells in the white blood cell, like use antibodies to fight against infections. And then the monocyte, they clean up the cells.
Is it my network?
Sister, please, can you hear me?
Yes, I can hear you.
Okay, so the monocyte, they clean up the dead cells from the system. And then in general, we say the white blood cells is part of the immune system. Thank you.
You're welcome. Any other submission? Any other? Okay, let me see. Where is Daniel? Daniel? Absent?
Madam, I'm there.
Yeah, can you share your idea on this?
Okay, madam, from the slide, as my sister said earlier on, we can see that we have two types of white blood cells, and these are the granulocytes and then the agranulocytes. That is the granular leukocytes and then the agranular leukocytes. And under the agranular leukocytes, we understand that we have neutrophils, which are responsible for if there is any bacterial infection in the body. It is these neutrophils that will invade that particular site in order to fight or eliminate the bacteria, or if there's a viral infection. And we also have the eosinophils. Eosinophils are also responsible if there is any allergic reaction. For example, if the body if there are some substances that come in contact with the skin, and there is allergic reactions, it is if those substances come in contact with the body, it is the eosinophils that will come, that will alert the body that there is something unpleasant to the body. So they will come around that site and then cause some reactions, so that the signs and symptoms of allergic reactions will be seen, and then maybe it will be dealt with by the body's own mechanism. Then we have the basophils, which also they are also responsible for some allergic reactions, but in this time around they are found, they are useful in the case of asthma. So if there is any asthmatic attack, it is these basophils contact to tell the body that there is something going on in the body. So the bronchial tree has to constrict and then produce maybe a lot of mucus in order to clear it. That is with the body's own mechanism, and these brings about the signs and symptoms of the asthmatic attack and the allergic reactions with regards to the asthma.
Okay. Well said. Now let's take this as an assignment. I have 160 students, or 164. 164 students in class today. Wait, just hold on. Let me take the attendance quickly.
Doctor, please, what is the question?
Oh, just describe the slide. No, you are doing it, but as clinicians, I wanted a lot from you. Okay, so for the assignment,
I want to also try.
Please give a clinical scenario, not a scenario, a clinical function of white blood cells. What I'm expecting is you can name any condition, specific condition of which any of these white blood cells play a key role in it. Do you get the question? So all I need is, yes, this is just, it's there, you are reading neutrophils first to respond to bacteria and all this. And you give me a clinical condition where you explain that neutrophils are doing this, that, same for the other type of white blood cells. So please do that. And let me quickly take the attendance so that I know those who are in class, those I'll be marking their work. Okay, so if you yes. Okay.
Madam, please, can we get the question again?
A minute, let me take shots of this and get back to you. Jenny, Jamila, Anilati. And next, Jadum. Eleanor Fulua and Lillian Solomon. Etc. Ebenezer. Dr. Ebenezer. All right, done. So the question is, give me a clinical condition it shouldn't be just, it can be any condition, okay, or any infection. And the process, not the process, the function of neutrophils in there, the functions of eosinophil in there, okay, the function of basophil in there, not just one. They should be unique to each of these white blood cells because they are all not doing the same thing. You get it? So I want just the function of these white blood cells, but use it in the clinical setting. I hope you get it now. Explain it in the clinical setting. All right. So basically we have the abundant white blood cell, which is the neutrophils, followed by lymphocytes. Lymphocytes, you have B cells, you have T cells, you have natural killer cells. All these are lymphocytes that help provide immunity to the body. After that, we have the monocytes, we have eosinophils, and then the basophils, which are less than 1%, which is the minute white blood cells in the blood. Then we look at platelets. With platelets, they are cell fragments. And then we are saying they do not contain nucleus just like the red blood cells. They are also produced in the bone marrow just like the other cells. And then when there's a blood vessel rupture or there's a cut, we are saying is this platelets and together with clotting factors that help blood clotting. But it's these cells that initiate the clotting process. So the cells would aggregate at the site of disruption, and then it will call for more platelets at the area. And then we also have the other clotting factors at the end, forming a fibrin. A fibrin is like a mesh that is formed at the wound where there's an opening to stop blood from leaving the blood vessel to the surroundings. I'm saying the clotting factor, the clotting process is also known as coagulation. And it's an important process to prevent excessive bleeding when a blood vessel is ruptured or is disrupted. We are saying that these platelets, together with some proteins called clotting factors, found in the plasma that work together to stop bleeding by forming a clot over the injury. Now, first of all, whenever there's a cut or damage to blood vessel, for example, this is an artery, a big artery, there's a cut, or not to even use an artery, a capillary, very small, tiny capillary, there's a cut, blood is coming out, let's say at the tip of your finger. We are saying the injury to the vessel triggers the release of clotting factors. Okay, and these clotting factors, as I said, is found in the plasma. Now, but the first thing that happens during during bleeding is blood vessel constriction. Blood vessel constriction. We are saying vasoconstriction would narrow that particular blood vessel such that there will be less blood flow to that area, so that you reduce the blood that will be leaving the capillary or the blood vessel out of the body. So the first thing that happens is blood vasoconstriction or blood vessel constriction. When that happens, it triggers clotting factors to move to the affected area. As that happens, we are saying platelets also moves to that area and forms a sticky plug. So that sticky plug helps activate more platelets to rush to the area. When that happens, we are saying we have the blood clotting factors working together to form a fibrin. Fibrin is a strand or mesh-like strand that would come around or wrap around this aggregated platelets and clotting factors, and that will stop bleeding. So what that you are saying is that when there's an injury, these clotting factors move to the affected area together with the platelets, and whilst the clotting factors move to the affected area, we have prothrombin, which is also a protein, being converted to thrombin. Then thrombin converts fibrinogen, which is soluble, to fibrin, insoluble. So it's this fibrin that forms a mesh-like strands around the aggregation to stop bleeding. As I said, these are the clotting factors. It's a whole process that for your level, you don't need it. So just know that we have clotting factors that would work together with platelets to stop bleeding. And with these clotting factors, we have factor 1, fibrinogen, factor 2, prothrombin, factor 3, we have tissue thromboplastin, calcium ions, lobar factor, stable factor, and so on. So all these work together to stop bleeding. But in a summary, we are saying in the presence of calcium, we have prothrombin converted to thrombin, and that is done by the prothrombin activator. Then this thrombin is also converted or converts the soluble fibrinogen, which is a protein, to an insoluble fibrin fibers. And these fibrin fibers form a mesh-like strands that wrap around that platelet plug or sticky plug to stop bleeding. Now, we have bleeding control mechanism. And this mechanism is what I've explained, but let's look at it in the flow chart. We are saying there's an injury to a vessel. Then we have constriction. So this is constriction of the vessel. And then when constriction happens, we have adherence of platelets and clotting factors, forming a sticky plug. And then we have activation of the other clotting factors that would enable fibrin to be formed at the end. And this fibrin would help clotting it will help formation of clot and therefore stop bleeding. Now, you are saying there can be prolonged bleeding. Okay, so when I'm sure those who work in the lab, they have bleeding time. So the assess saying a bleeding time. So if the person's bleeding time is not normal, you are saying there can be as a result of low platelet count. Okay, so the bone marrow is producing enough or normal volume of platelets. We are also saying that we have inadequate platelet function, where there are platelets all right, but they are insensitive to rupture of blood vessels, or they can also move to the affected area all right, but maybe there's 1 or 2 instances where they still can't perform their function together with the clotting factors. And also when we have inadequate constriction. So a constriction is when we are saying that the vessel cannot narrow because there's maybe low calcium levels. When there's no calcium levels, there's less contraction of the muscles, blood vessel muscles. When there's less contraction of blood vessel muscles, meaning the blood vessels can't constrict well. When they can't constrict, meaning when there's a cut, blood will still ooze out, blood will still come out because the blood vessel can't narrow. And all these are what can cause prolonged bleeding time. Then we look at a common clinical test that is done in the lab. And this also speaks a lot about the condition of your blood, okay? And we are saying complete blood count or full blood count is a common diagnostic test that is done in the labs to help with diagnosis, okay? So full blood count together with other tests help clinicians to diagnose any malfunctioning in the blood or any components of the blood, and even it informs clinicians about other conditions. We are saying complete blood count is made of red blood cell count. We have hemoglobin, we have hematocrit, we have red blood cell indices, white blood cell indices and differentials. We have platelet count and its differentials. So all these are there to ensure that indeed the level of red blood cell in your body is normal, and the homeostasis, the normal level of white blood cell is right. You have normal levels of platelets, the normal size of red blood cell. We have the normal weight of red blood cells, normal volume of hemoglobin, normal concentration of hemoglobin. All these parameters are there that enables us to assess whether the blood is in its normal state. So as I said, red blood cell, we are counting the number of red blood cells. When you take a microliter of blood, what is the normal number of red blood cells inside? That is the red blood cell count. We are saying we have hemoglobin concentration. We are looking at when you pick one red blood cell, within that red blood cell, what is the concentration of hemoglobin in there? And look at hematocrit. Hematocrit, I mentioned that is the percentage of red blood cell in the blood. So hematocrit is looking at the percentage. Red blood cell count is looking at the number. Okay, so the number of red blood cell. Then we look at red blood cell indices. So with red blood cell indices, we have the MCV, the mean cell volume. We have MCAC, the mean corpuscular hemoglobin concentration. We have others, okay. Then we have white blood cell counts, looking at the number of white blood cells and its differentials. So the differentials are the different types of white blood cells we have, and their counts, and their percentages. Platelet count, we are looking at the number of platelets differentials. We are also looking at the normal volume of platelets, the mean concentration of platelets, and so on. So all these help in making sure that indeed your blood is in a normal state, whether it's volume of these components and all that. Then we look at how blood is formed. How blood is formed. What time am I in the class?
Very 30, please.
3:30. Hello?
Yes, honey.
All right.
3:30. No fire. No fire.
I think 3:45. Someone to say 3 p.m. Hey. So I'm ending at 3:30. I was supposed to start at 1:30. Yeah. All right. So the process of blood cells formation is called hematopoiesis. Hematopoiesis. We're saying hematopoiesis happens in the bone marrow. And in the bone marrow, we said we have some cells called stem cells. And these stem cells, as I said, is within these cells, or is these cells that will differentiate to form different types of cells. And we mentioned earlier that is these stem cells that will differentiate to form the red blood cells, the white blood cells, and the platelets that we have. So with the hematopoietic stem cell, I made mention that it differentiates into two main progenitor cells. And this is the myeloid progenitor cells and the lymphoid progenitor cell. It's these two cells that differentiate into the different cells that we have. So the myeloid progenitor cells is going to differentiate into the red blood cells, into the monocytes, the neutrophils, basophils, eosinophils, and platelets. And then we have the lymphoid differentiating into the lymphocytes. The lymphoid progenitor cells will differentiate into the lymphocytes, only the lymphocytes. The rest of the cells are differentiated from the myeloid progenitor cells. Now, how is blood production initiated? Now, since we know that red blood cells is the abundant cells in the blood, it's when there's less supply of these red blood cells, or when the function of this red blood cell is altered, that will trigger production of red blood cells. I don't know if you get me. Whenever the red blood cells function is limited, meaning the body interprets is that the red blood cells in the blood is reduced, and therefore we need to produce more. And we've already stated that the red blood cell functions to transport oxygen to every cell in the body. And therefore, if we don't have enough oxygen reaching every cell, then the body signals the bone marrow to produce red blood cells. Now, when we have less oxygen reaching the tissues, we term it as hypoxia. And this blood circulation would reach the kidney as well. So in the kidney, we are saying we have production, a production of a hormone called erythropoietin. This erythropoietin is produced from the kidney. It travels back into the blood, travels through the blood to the bone marrow. And it's within the bone marrow that this erythropoietin will trigger the hematopoietic stem cell to be differentiated into the myeloid and then the lymphoid progenitor cells. Then the myeloid cells would also differentiate into red blood cells as well. Red blood cells will be released into the blood to increase the number of red blood cells in the blood. So we are saying conditions that causes low tissue oxygenation. Number one, when you have a reduced blood volume. When you have a reduced blood volume, whereby you are not taking enough water, and therefore your blood plasma is reduced, that can signal the system to produce more red blood cells. And also, when we have lower number of red blood cells or less hematocrit levels, we are saying, or percentage, we are saying that will also trigger the kidney to produce erythropoietin and then go through hematopoiesis. Then, as I said, anemia, specifically when you have low levels of hemoglobin. So if the hemoglobin inside the red blood cell is not enough, is not normal, it's going to trigger the production of red blood cells. How? Since the hemoglobin in the red blood cell is not enough, it's not picking the needed oxygen from the lungs. And therefore, we have less oxygen reaching the tissues and the cells. Therefore, the body interprets that as less red blood cell count. Also, when there's something wrong with the heart, and therefore it's not able to pump the needed volume of blood out to the body, and therefore when there's blood flow, low blood flow in the system also interprets it as lower or less red blood cell count. Therefore, produce more red blood cells. Or if there's any problem with the lungs, okay, any problem with the lungs, any part of the lungs, and mainly when there's perfusion, okay, at the alveoli level, where there's exchange of the oxygen and then the blood, we are saying that would limit then the concentration of oxygen diffusing into the blood. Therefore, oxygen would not reach the cells. Therefore, the body interprets that as lower number of red blood cells. All these conditions would lead to the production of red blood cell. The production of red blood cell is known as erythropoiesis. But since we are saying the red blood cell is 45% whole blood, it's the process of erythropoiesis that also helps with the formation of the other cells. So with erythropoiesis, we are saying it happens in the presence of erythropoietin, which is produced from the kidney. We have in the presence of iron, vitamin B12, interleukin 3 and 4, and others that help in the development and maturation of red blood cells. Now, we have the hematopoietic stem cell. And this hematopoietic stem cell in the bone marrow, in the presence of erythropoietin, will differentiate into the pro-erythroblast. That's from the myeloid progenitor cell. It differentiates into pro-erythroblast. Then this pro-erythroblast also differentiates into erythroblast. During this process, we are saying the cell starts to lose its general structures that the cell has. So it starts to lose its organelles, it starts to lose its not generalized shape and all that. Then when it gets to from erythroblast to reticulocytes, you're saying most organelles are lost, including the nucleus. So we have most organelles are lost, including the nucleus, at the transition from erythroblast to reticulocytes. Reticulocytes, some are released into the blood as reticulocytes. So you have about 1% of circulating red blood cells, which are reticulocytes. Then when they move into, they are released into the blood at that stage, at the reticulocyte stage. When they get into the lungs, they mature. Sorry, when they get into the blood, they mature. During maturation, we are saying they lose all other remaining organelles found inside the cell. And at that point, we are saying that is the matured erythrocyte or the red blood cell. The lifespan of red blood cells is 120 days. And therefore, when it gets to the 120 days, it moves back to the liver for breaking down of these red blood cells, and then for new, and it triggers new formation of red blood cells. All right. So we are saying that the hemoglobin that we made mention of, that is found in the red blood cell, its synthesis or production starts from the erythroblast. And from the erythroblast, we are saying it starts as basophilic erythroblast. It then differentiates into the polychromatophilic erythroblast. Then it finally moves to the autochromatic erythroblast. So all these, this is a summary of it, because if you have to take it step by step, we have other steps that we skip. But in summary, we are saying this is the process of red blood cell production and maturation. So in the bone marrow, we have the hematopoietic stem cell differentiating into the myeloid progenitor cell. This progenitor cell will differentiate into pro-erythroblast, then to erythroblast, then to reticulocytes. The reticulocytes are released into the blood, and then in the blood they mature, losing all other remaining organelles, forming a matured red blood cell. Okay, so mind you, when we have the differentiation of hematopoietic stem cell into the myeloid progenitor cell, we have the other cells taking up a different pathway, forming the other different types of white blood cells and platelets. Okay, but with the red blood cell, we are saying the progenitor cell differentiates into pro-erythroblast. Anemia. Anemia is caused by a lot of factors. So the first one is hypoxia, as I described. So we have decreased oxygen supply to the tissues. So when the tissues and cells are not getting enough oxygen, we are saying that can be classified as anemia. Okay, and this anemia, you can have instances whereby is the low level of red blood cells. You can have instances whereby it's low level of hemoglobin. You can have instances whereby there is blood loss and so on. Okay, so anemia is caused by different factors. So the second one is decreased red blood cells, as I said. So the number of red blood cells are decreased. Therefore, oxygen is not supplied at their normal levels. Decrease hemoglobin, as I said, and then decrease hematocrit. So you can have types. The first one is hemorrhagic anemia. Hemorrhagic anemia is when you have excessive loss of red blood cells through bleeding. Okay, bleeding from any part of your body. And as I said, bleeding is mainly from disruption of the blood vessels. Aside that, you are saying when you have conditions such as stomach ulcer, and then the females as well, menstruation. Menstruation, we are saying bleeding from menstruation is made of the uterine walls shedding off together with the normal blood flow. Okay, so as women, we tend to have lower levels of red blood cells and even less percentage of red blood cells compared to men due to this. Then we also have hemolytic anemia. So we have hemolytic anemia. With hemolytic anemia, we are saying we have instances whereby red blood cells the membrane of the cells ruptures. And that can be as a result of infections, parasites, toxins, and the common infection that we all know of that causes reduction in red blood cell is malaria infection. So these parasites invade the red blood cells and then ruptures the red blood cells. Therefore, you tend to reduce the number of red blood cells found inside the blood. Then we can also talk of thalassemia. Thalassemia is hereditary. It's a condition whereby you have less synthesis or production of hemoglobin, and it's genetic, and it's found in populations of the Mediterranean Sea. And you can have one or two conditions around where there is production of red blood cells, all right, but within the red blood cells, we have few hemoglobin in there, or the concentration of hemoglobin in the red blood cell is not normal. Then we have the commonly sickle cell anemia, where that we are also saying that is the hereditary condition, and it's characterized by red blood cells that assume an abnormal rigid sickle shape. And their shape gives them an abnormality as to the function of this red blood cell. All that we are saying is that the sickle shape reduces the hemoglobin concentration inside the red blood cell. And therefore, that also limits the number of oxygen that can be transported by the cell. Also, the shape is also abnormal, and therefore, there can be instances whereby it can block blood vessels. And you have aplastic anemia. Aplastic anemia is when you have destruction of the red bone marrow where there's production of blood cells. That can be caused by radiations mainly. Radiations, we also have toxins that comes in there. So basically anemia can be caused by a lot of factors. Blood grouping system. A minute. Okay. All right, so let's end the class for today. When we meet next week, we look at blood grouping, and then erythroblastosis, and then look at compatibility, and then we look at resource. Basically, that's that. We can use an hour to finish that. Then we end the class for next week. Any question? Any question at this point? Any question?
No, madam.
Hey, today the class is quiet too. Or I should give the slides to you. You know everything. I'm sure you know everything.
Oh, madam.
No. But today you are very quiet.
We are struggling to cope.
Come dear.
People are tired.
You are tired. I know, right? You are tired.
It's clear, so we are okay.
It's clear. Yeah.
Yes, it's clear, madam. It's clear.
Yeah, but you are doing the anatomy part, right?
Yes.
We enjoy
Yes.
that.
Okay, that's fine. You'll be saying it's clear, but at the end of the exams, then I don't see that clarity.
Madam, please, can we get a slide?
Oh, yeah, I'll send it right away.
Okay.
Yeah,
Madam,
so
Madam, if the exam is not clear, then
will trick us during the
it will be clear. Let the question be.
So where is mine? So with the end of semester exams, with the introductory part, are you okay? Because I've assessed you on that. So if you are not okay, I think looking at the performance, it wasn't the best though, but I wanted to assess you on the nervous system and then the cardiovascular system for the end of semester exams.
Wow.
Since I've assessed you already on the introductory part. If you are not okay with the introductory part, I can still bring in that part as well. But if you are clear on introductory, then let's expect the nervous system, and then the cardiovascular system.
Okay, madam, thank you very much.
So with the nervous system, in fact, every part of it, from the divisions and the functions and all that, coming down to action potential and all environs of action potential, the homeostasis. So these three main components. So it's going to run across. Then with cardiovascular to where we end next week. Okay.
Okay.
Theory energetics.
Come again.
Is it theory and objective?
For that one, it's MCQs. It's MCQs. MCQs. I tried adding about two session B's on written. But let's see. I'll update you on that next week. On my decision.
Okay, thank you.
Okay.
Yeah. So we end the class for the day.
Madam,
Yeah.
please, is it possible we can see our midterm results so that we will set up well?
I did that for the two groups I taught last year, but some objected. Okay. Because some people will look at other people's marks and all that. So if you want to see, just message me privately. I'll let you.
Okay, madam.
Yeah.
Thank you.
You're welcome.
Madam, how do we
Yeah.
submit the assignment you're giving?
The assignment is a reading assignment. It's a reading assignment. We will discuss. Yeah, we will discuss it next week. Next week we have more time. We'll have a discussion. And in fact, that part is coming. That's why I gave it to you as an assignment. It's better you go and read. Okay.
Yes, ma'am.
Most of the questions are not just the way I've taught it. It's related to the clinical setting. So it's application. You get it. So you have to prepare very well. I hope that is clear.
Madam.
Yes,
Hello,
ma'am.
madam. Please, some of us, due to network issue, couldn't take part in the midterm.
Yeah, I only had 3 people, and that is all. I had 3 complaints. So, and I learned you are over 300. But today's class, the number of people who came, 180, I saw 184 somewhere during the teaching. So let's say 180. So we have about half of the class not joining. And this is something that runs through since the beginning of every class. I don't know if it's only physiology. But other class you attend.
I think we are like 200 and something, madam. We are not up to 300.
The number of students who submitted their midterm, there was 269. And those of you who submitted twice, I don't know if someone submitted twice with the same name and the index number, but the person used different phone or laptop. Is the first one that counts is what I'm going
Madam,
to use.
you can bring it and omit the names so that we can see with our index numbers.
Okay, that's fine. So you want that way. So you want just your index numbers and your mark.
Yes, please.
Okay, then I'll do that way.
Madam.
Hello, madam.
Madam, please, the person you are talking of that said they use a different phone. I think I'm the one because the first phone went off, which I didn't complete. So I have to use a different phone.
Okay. No, then it's not you because the person I'm talking about submitted. You get it? If it went off, that means you didn't submit.
Really?
Yes.
Yeah, madam. Madam, it's me.
It's you. It's you. So it's the first report that I'm going to use. The first one.
Yes, please. I think it was a mistake. It was a mistake. I sent you a message and I'll tell you that.
No, it's not that one. It's not that one. Don't worry. But the main reason why I prefer you look at your marks is not really your marks, but your index number to make sure that it's correct. Because at the exam surface, we have a lot of issues, even currently a lot of issues, whereby you have challenge with writing your correct index numbers. You get it? So when I send it to you, kindly cross-check your index number if that is right. and make sure you shade well in exams. Otherwise, it will be only your midterm or your end of semester that will come, and it can cause trails. So your index number, getting it right is very important. Okay. Enjoy your evening.
Thank you, ma.
Thank you, ma.
All
right. Bye.